Hi everyone. In about 5 minutes, we'll get started with Woods Hole Oceanographic Institution's Jellies, the surprising role of gelatinous creatures in ocean and human health. We've still got a lot of people joining us, so thank you for your patience. Hello, you've joined Jellies, the surprising role of gelatinous creatures in ocean and human health, a Woods Hole Oceanographic Institution Ocean Encounters presentation. We have a whole lot of people joining us tonight, so please bear with us. We'll get started very shortly. The ocean covers 70% of the globe. It gives us oxygen and food and millions of jobs. It brings joy and shapes our climate and weather. The ocean is life.
and it belongs to everyone. Woods Hole Oceanographic Institution is the world's independent leader in ocean discovery, exploration, and education, working to understand and sustain one of humanity's most precious common resources. Join us today for our ocean, our planet, and our future.
Welcome to Ocean Encounters, a series from Woods Hole Oceanographic Institution, or WHOI as we like to call it for short. Tonight's event is Jellies, the surprising role of gelatinous creatures in ocean and human health. My name is Veronique LaCapra, and I'll be your host for tonight. WHOI's Ocean Encounters events are made possible in part by the Avatar Alliance Foundation and Dalio Philanthropies. Thank you. Our fifth season of Ocean Encounters is also an endorsed activity of the UN Decade of Ocean Science for Sustainable Development. Before we hear from our panelists tonight, I'd like to take just a minute and share some tips on how you can optimize your experience with us on Zoom. We'll be taking questions from all of you throughout the evening for our panelists. If you'd like to participate in that live Q&A, please use the Q&A button on your Zoom screen and type your question in the window that appears. You might be more familiar with the chat function in Zoom, but for tonight, please use the Q&A function instead. We often get hundreds of questions, so I apologize in advance if we don't get to yours while we're live. You can ask questions anytime, starting now. I also want to let you know that we are recording this event. That recording will be made available on the whoi.edu website. We had more than 4,300 people pre-register to join us tonight, so you are in very good company. We also have viewers joining us right now on Facebook and on YouTube, so to all of you, welcome. Tonight we're going to talk about jellyfish and other gelatinous ocean animals. Jellyfish may be best known for their toxic stings, but they also play an important role in ocean ecosystems, from seagrass beds to the deep sea. Tonight, we're joined by three biologists who study jellyfish in a variety of ways as part of their research. We'll hear how they do that, the techniques and the tools they use, and also what jellyfish can teach us about the health of the ocean and human health, too. Please join me in welcoming JOCELYN MALAMY, ANNETTE Govindarajan, and Larry MADIN. Annette and Larry are both from WHOI. Annette is a biologist and an oceanographer. Larry, a marine biologist specializing in gelatinous ocean life. JOCELYN is a cell biologist from the University of Chicago. She's also a Whitman scientist at the Marine Biological Laboratory, one of WHOI's neighbors in Woods Hole. Welcome, everybody. I'm not seeing Larry. Are we having technical difficulties, maybe? There we go. Great. Hi, Larry. Tell us a little bit about jellyfish, Larry. Are they basically what we picture, translucent and bell-shaped with tentacles?
Well, actually, they're all different sizes and shapes of jellyfish. When we talk about jellyfish, we mostly are referring to what we call medusae, which are the little round or saucer-shaped things that often can sting people. These live all throughout the world's oceans. There are more than 3,000 species of medusae. So that's the common concept of what a jellyfish is, and a lot of what we'll be talking about tonight. There are a number of other kinds of gelatinous animals, and I think we'll get to some of those in the course of our discussion.
Oh, I'm sorry, I was on mute. I was saying that you've studied jellyfish for a really long time, I think over the course of your 45-year career, I believe. What got you interested in them?
Well, actually, it wasn't jellyfish per se that I got interested. When I was a graduate student, I learned about salps, which are in one of the other gelatinous animals we'll talk about tonight. And they're animals that form long chains, as you see in the video here. They're completely unrelated to jellyfish, but they're really important in the ocean because they're filter feeders that take in the small algae cells and they turn them into larger fecal pellets, which we often call poop. And they're playing a really important role in the economy of the ocean today. I was really fascinated because very little was known about them and they'd never really been studied in the wild. You get an impression from this video of what these beautiful chains of animals look like.
What other kind of gelatinous animals are there?
Well, they're actually gelatinous representatives of many of the different animal groups in the ocean. This one is a ctenophore or comb jelly. They don't really have common names except comb jelly. And as you can see from these pictures, they're beautiful to look at. The rippling colors come from the diffraction of light on the little vibrating cilia that they use to swim with. So these occur also throughout the ocean, not nearly as abundant number of species, and they're mostly predators on smaller animals, smaller crustacean shrimp-like animals.
So that light we're seeing is actually natural light, bioluminescence as it's called, that they are producing themselves.
Well, the brightly colored light is actually sunlight or artificial light that they refract into rainbow colors.
Oh,
but
wow.
You make a good point, because they are also very brightly luminescent. It's harder to see that in the photographs because it's not as bright.
All right. So why are there so many different kinds of gelatinous animals in the ocean? Are there advantages to being squishy and see-through?
Well, I think the main advantage that they have is, as we saw in the beginning of the show, that their bodies are about 98% water and only a couple of percent of actual sort of living tissue. This enables them to be much larger than they would otherwise be for the same amount of biomass. And because they're larger, they can either evade predators by being too big to eat, or they can be better at eating other things because they can eat things that are smaller than themselves. They're also neutrally buoyant in seawater, so it's not as hard to swim. And they're transparent for the most part, so it makes them harder to see for a visual predator like a fish.
What are we looking at there?
Well, this is one of the longest jellyfish. This is actually called a siphonophore. It's a relative of the jellyfish made up of many, many different units. This one is estimated to be well over 100 meters long. And it's an example of how these animals that are put together like a colony of many, many different individuals can reach extraordinary size. This is the way these siphonophores, which also catch prey like jellyfish do, can achieve this kind of a size in the ocean, especially in places at depths where there's not much turbulence, because this would easily be disturbed if there were waves crashing over it.
That's amazing. All right, let's see if we have any audience questions yet. My colleague Ken Costell is behind the scenes fielding audience questions tonight. Ken, have you got a question for us?
Oh man, they are coming in hot and heavy, but on Zoom. But I'm going to turn to Facebook for our first question. Kelly asks, are they affected by light pollution? And I guess I would modify that a little bit and say, are jellies sensitive to light?
Larry, you want to take that?
Well, actually, yes, some of them are. Some of the jellyfish actually have little light receptors around the edge of the bell that we call it. The salps that we saw earlier have an eye that's basically right in the back of their head, and many of the other gelatinous animals that fall in other groups, like mollusks and worms, also have eyes. So they are sensitive, and some of them. There's a type of jellyfish that even has an image-forming eye, which is quite unusual for such a relatively simple animal.
Wow. All right, I want to turn now to some of the connections between jellyfish and human health. And probably the number one thing, as I said earlier, that people know about jellyfish is that they sting. Annette, you study clinging jellyfish, which can pack a powerful sting. Tell us a little bit about them.
Right. So clinging jellyfish are very small jellyfish. You can see them there. They've got on the screen, they have these sticky pads towards the ends of their tentacles that allow them to cling to eelgrass or seaweeds or to the side of the aquarium wall, like you can see here. And so they're, like I said, they're small, dime to quarter size, but despite their small size, they can cause stings that in some people can cause a lot of pain. And so so it's very interesting. We have them here in the Cape Cod area, and we started noticing some years ago that we started getting reports about people getting these really nasty stings. And so we'd had these jellies in the area, but hadn't had reports of the sting. So we were thinking we were experiencing something new, and that perhaps these a new strain may have potentially been introduced from another part of the world where the stings had been recorded.
So we saw some clinging jellyfish there in a petri dish a moment ago, so they can really be quite small, and they don't look like they could swim very far to me. So how would they suddenly appear in a new location on the other side of the world? How would they get there?
Yeah. Oh, that's a great question, because you're right, they're not really good swimmers. And so to understand how they can be transported long distances, we look to the whole life cycle, right? And so the jellyfish that we see pulsing on that dish, that's just one stage. And so the jellyfish reproduce, they produce larvae which settle and form microscopic polyps. And you're looking at some actually sticky settled eggs on a piece of eelgrass there. And these polyps then they, through an asexual process, produce medusae, or the jellyfish, right? And so these are some microscopic polyps. So they're very tiny. This is looking at take a picture taken under a microscope. And so these polyps can live on different surfaces, such as bivalve, like oyster shells, right? And so if we're moving around the oysters, right, those little polyps accidentally can be hitchhiking along with that, right? And so that is one way which these kinds of animals can potentially be transported long distances.
So they're attached just like a little sea anemone or something like that?
That's right, except that you wouldn't even notice them because they're so tiny, right? And so these pictures were taken with a microscope. So you wouldn't do it, you wouldn't even realize that they were being transported.
JOCELYN, like Annette and Larry, you do research on jellyfish, but for kind of a different reason. You're actually studying them to learn something about human health. Tell us about that.
Yeah, I can tell you about two of the projects that are going on in my lab. We work on a little jellyfish about the size of your thumbnail. It's called Clidia hemispherica. You can see it swimming around there. And we got excited about it when we started looking at it under the microscope. And when we look down on the top of the jellyfish, on the top of its bell, you can see its cells. And those hexagonal cells, they're called epithelial cells. It's sort of like the skin. And those cells, if I showed you our own cells from our lining our guts or our lungs or on the surface of our cornea, they would look exactly like these cells. So they have these cells that look just like ours, and we can easily wound them just by making a little scratch on the surface of the jellyfish. And what's really
hang on a second. So wait, just to orient people, so those going back to the previous one, if we can. So the little hexagons in the bottom half of the picture there, those are individual cells that are just from magnifying the surface of the jellyfish. Is that what we're looking at?
Right. That's right. We're looking at the surface of the jellyfish under a microscope, and each of the hexagons of the cell, and you can see a nucleus inside each one of those cells.
Little bumps in the middle.
Yep. And then on the panel right next to it, you can see those same epithelial cells, except now there's a wound in the middle. And so you're looking right down on the jelly of the jellyfish, and some of those epithelial cells, those skin cells, aren't there anymore.
The blank part.
Exactly.
Okay. So now let's continue on.
That's fine. So this is a little time-lapse movie where we're taking a frame every 10 seconds. And what we can see is each one of those cells is participating in healing that wound. And they do it really, really well and do it really quickly. So you can almost see these healing before your eyes. This whole movie is around a 45-minute movie.
That's really amazing. Incredible.
So sorry, go ahead.
No, go ahead.
And so this is the kind of thing that we can't really see if we were to look at a wound in ourselves. So the ability to see this, at such high resolution, these small cells and exactly how they move in a live, intact animal, is really special and something that jellyfish is a kind of unique system for that. And we have another project.
Go ahead. Anything else? Yeah.
Yeah. So the other project that we work on in my lab, it's a collaboration with a professor named Eduardo Perozzo, and he's interested in a special kind of cell. It's called a hair cell. And we have hair cells inside our ears, in our inner ears, and they have little projections that are sticking up. And when sound waves come into our ears, these little projections get deflected. And the deflection of those little hair-like projections are what help us to hear. They send a signal to our brains that help us to distinguish sound. And jellyfish also have hair cells. Their hair cells are located on their tentacles and around their mouths. And those hair cells are triggered not to give you a nerve impulse, which goes to a brain, but when you trigger those hair cells, they actually shoot at you and they sting you. And that's how they sting. And Dr. Perozzo, his idea is that these hair cells in our inner ears are very similar to these hair cells in jellyfish. And so if that turns out to be true, the ability to see them so well in jellyfish and to be able to get our hands on so many of them, because of course jellyfish tentacles are just covered with stingers, would be a very important advance. And so you're looking here at a picture on the left of a hair cell from a mouse inner ear and a hair cell from the tentacle of our jellyfish, Clitia hemispherica.
Yeah, they look really similar. That's neat. What other kinds of research is going on with jellyfish related to human health or basic biology?
Well, jellyfish have actually given us one of the key tool for cell biologists and developmental biologists. It's something called green fluorescent protein, which we abbreviate as GFP. And it was originally isolated from these jellyfish. They're called, and someone can correct my pronunciation, Aquaria victoria. And Aquaria is a jellyfish. It kind of glows blue, but it also has a green glow. And that green, you can see it in these little dots around the margin of the bell. And that's caused by this protein called green fluorescent protein. And it was first isolated by Dr. Shimomura, who at that time was an MBL researcher. And when he isolated this protein and came to understand it, what he and his other colleagues realized is that you could take this little green fluorescent protein and stick it like a tag onto other proteins. And so all of a sudden, that made proteins that we were interested in watching in all kinds of cells and all kinds of animals, something that we could see. So all of a sudden, you could watch the proteins that you were interested in live animals. So this is a particular protein that is in a zebrafish spine. And this animal is alive, and we can see what's going on inside this zebrafish. Or we can actually zoom in on individual cells. In this case, there's a protein called actin, and we've made it green by adding a green fluorescent protein to it. And so if I had a movie of the cell, you'd be able to see what this protein was doing while the cell went about its business of doing all sorts of things that these cells do.
That's both fascinating and beautiful, I must say. Any other applications you wanted to mention?
So there's another really fascinating lab at MIT. So my colleague, Brady Weisbord, he actually got converted to studying Clitia hemispherica when he saw them growing at the MBL. And he's a neuroscientist. And in his lab, he studies perceptions and responses. And because jellyfish like Clitia are so small and they're transparent, I think we can see a picture from his lab somewhere here. Because they're so small and they're transparent and their behaviors are regulated not by a central nervous system, they don't have a brain, but by a net of different nerves. And in this image, what we're seeing is the firing of each of the nerves. So he's used a technique that allows you to see those nerves glow when they fire. And that allows him to understand the neural networks that regulate an entire behavior in this animal, in this live animal.
So it would be like if we were to do some sort of scan in our own brain to see the neurons firing, but you can actually just watch it here on this video.
That's right. I mean, of course, our brains are much more complicated than those of a jellyfish, but still, our understanding of how you stimulate a brain and that stimulation gets turned into an action or a response is still very, very rudimentary. So being able to understand that in a simple animal is maybe a more tractable problem, and we still have quite a lot to learn, even from a simple animal like a jellyfish.
Let's take another question from the audience. Ken, what do you got for us?
Well, we have over 100 questions already. So first one from Claire for Annette. How do jellies give birth? Do they all go through that same process that you described for clinging jellies?
Yeah, so that's a good question. Jellyfish life cycles Can be complex. So the jellyfish that we think of as a jelly, that's actually the adult stage. And so you can have males and females, and they reproduce and form the larvae, right? And then the larvae are what settle and form the polyps, or sometimes they're called hydroids, and then they can, through an asexual process, then produce the jelly again, right? And so that's kind of the typical life cycle, but there are variations on that theme where you might have a species that lacks the jellyfish stage entirely, or that it's, it may lack the polyp stage entirely. So there's all kinds of variety, and that's one of the things that I think makes jellyfish very interesting to study.
And one other, I guess this would be for JOCELYN. Abigail asks, how thick is that layer of epithelial cells? And I would add then, does that healing occur the same across the depth of that layer?
Yeah, so that's one of the nice things about the Chlamydia system. So the skin that we're looking down on the jellyfish, it's only 1 cell deep. And those cells are big, flat, pancake-like cells. So they're only 1 to 2 microns deep, and they're 50 microns across. So what you're watching is just these individual, is groups, a sheet of these big, flat cells. And each one of those cells at the edge of that wound is moving across that gap and looking for each other, grabbing onto each other to close that gap and heal that wound.
So how magnified is that image that we're looking at now?
This is about 125 times magnified.
Okay. All right, Larry, going back to you, we've been talking about jellyfish and human health, but what about jellyfish and ocean health? How important are jellyfish to ocean ecosystems?
Well, I think we're really beginning to realize now just how important they are. I think for a long time, because they're squishy and soft and so forth, hard to sample, people didn't think of them as being as important as fish or shrimp and other things that we are more familiar with and that are easy to sample. But we do know that jellyfish, particularly the medusae we've been talking about, are important predators. They eat a lot of different things in the ocean. They themselves are eaten by things, even things as big as turtles and giant ocean sunfish, for example, that are predators on jellies. Some of the other jellies of a different sort, like the salps, for instance, are important because they mediate the flow of carbon from the surface layer of the ocean down into the depths of the ocean. So the more we're able to study them and see what they're doing when they're living, the better we are able to appreciate the role that they have in the food webs throughout all depths of the ocean. Yeah, they're also often, here's a picture of one catching a shrimp, for example. They also can have a lot of different sort of hitchhikers. And sometimes there are little animals that live inside the jellyfish or on the surface of them, where they are protected because of the jellyfish from other predators. So there's an example with this one we're seeing here, where the big fish is trying to get out the little fish that are hiding up inside the jellyfish. And so even though the jellyfish sometimes eat fish, they can also provide a refuge for them because they're a big globby piece of something to hide in the ocean.
Neat. So Larry, you touched on the relationship between jellyfish and climate. And Annette, I know you're involved in a project about the ocean twilight zone, which involves looking at the role of the ocean and the animals in the ocean in regulating climate. Tell us a little bit more about the role of gelatinous animals in that cycle.
Right. So gelatinous animals can play a role in bringing carbon down to the deep sea. So they're part of this what we call the biological carbon pump. So we have carbon dioxide in the atmosphere that gets taken up by phytoplankton or algae in the ocean, and then that carbon is incorporated in that biomass, and that gets eaten and by animals, and it moves through the food chain, and ultimately that gets transported down into the deep ocean. So jellyfish and gelatinous animals more broadly are part of that process. And in particular, there's a group of animals called salps, right? And I think Larry's there, we see them, and Larry's talked about them. And salps are filter feeders, and they can feed voraciously on a wide range of particle sizes. And so they do that, and they take up that carbon in the particles that they're eating, and then they produce these fecal pellets that they excrete. And they're relatively large fecal pellets and dense, and they sink, right? So by being consolidated into those fecal pellets and then sinking down to the deep sea, they're part of that carbon pump.
And we were looking there a moment ago at a video of what's called marine snow, which would include those fecal pellets and other particles that have a lot of carbon on them. So Larry, I wanted to go to you again, talking about, again, ocean health and climate. There's been quite a bit of, you might say, alarmist press about the idea that climate change and a warming ocean are driving massive increases in jellyfish populations, and that they're basically taking over the ocean. Is there any truth in that?
Well, I think that certainly we're seeing that the climate is causing changes in the ocean in a lot of places, in its temperature, in its chemistry, and its circulation. And one of the things that we have noticed is that there are populations of jellyfish that sometimes are larger than expected, or that appear in places where they weren't before. Part of this is because they do tend to have boom and bust cycles because of the nature of the reproduction. Another thing is that they may be moving into waters which are warmer than they used to be and are more hospitable to jellyfish, or waters that have less oxygen and are less hospitable to fish and other creatures, so that jellyfish kind of move in and take over the space because there's no competition. But we do know that over the long term, there's evidence that there's a cycle of boom and bust to many of these jellyfish. Whether it is a wholesale takeover of the ocean, I think is probably premature to say that that's going on now.
Is there a seasonality to jellyfish also at sea? I mean, just as a swimmer in the ocean, I feel like sometimes I see quite a few jellyfish, and at other times of the year I don't. Annette, is that related to what you were talking about with their life cycle? Where did the jellyfish go?
Right, that's another really good question. And so when we see the jellyfish, we're only seeing that's that one piece of their life cycle, right? And so they can be persisting in their polyp stage, and some of them have these stages where they can be dormant. So they're kind of there lying in wait, waiting until the environmental conditions are right for them to start then producing the jellyfish. And that's when we may see the bloom, with the right environmental conditions, and they're triggered to all start producing the jellyfish very rapidly.
That footage is
many
just
of these
so
large
incredible.
jellyfish have a lifespan of about 1 year or less.
So the jellyfish we're looking at now might only live for a year.
Yeah.
Wow. Ken, let's take another question from the audience.
Okay, let's see. Let's see, we have a lot of people are asking just how long do jellies live? Not just those that are there that you say might be a year old, but what's the longest lived jelly?
Or maybe gelatinous animal.
Gelatinous animal, thank you.
Well, I'll take a stab at that. We don't really know in most cases, because, and the reason we don't know is that most of these jellyfish, nobody has been able to keep them alive in a culture situation long enough to determine what their lifespan is. Now, for a little one like the ones that JOCELYN is using, probably we can tell that. But for a lot of these bigger ones out in the ocean, either we know from the change in their population from 1 year to the next what their lifespan probably is, but the ones in the deep sea or the ones further away, we're really not sure. Some of the siphonophores, which keep on growing and growing, could be tens or even hundreds of years old. We just can't tell from those guys.
Wow.
And one more, we have several people who are asking about blooms as an indicator of potential problems in the ocean. Are blooms always a sign of a problem, and how are they able to group together in general?
Some of the jellyfish remain together partly because of their swimming behavior, and if they are mostly swimming up and down repeatedly, they're not going to spread apart horizontally, and that tends to keep a group of them clustered together. The same is true for salps, which form large blooms like we saw in that earlier video. In fact, off our coast here in the North Atlantic, off of the east coast of the United States, there's a summertime bloom of salps that occurs pretty much every year. And that we get very high densities, but they maintain that also by mostly swimming up and down so that they all stay together in a cluster. Whether or not they have a sensory capability to do that is unclear in some cases.
Let's talk a little bit about the tools and techniques that are used to study jellyfish. JOCELYN, I'll go to you first. You study jellyfish in the lab. Are there any new technologies that have made your work possible or enabled it to advance?
Yeah, absolutely. I mean, so for a long time, cell biologists, developmental biologists, we really focused on a very small number of model systems that were easy to study in the lab, things like fruit flies, things like little C. elegans worms, things like yeast and mouse. And these are wonderful model systems. We've learned a lot about those animals and they help us understand ourselves, but we've always realized that they don't represent the entire diversity of life on our planet. But it took so much resources and so much work to understand what all the genes were, for example, in a fruit fly or in a yeast, that a lot of labs would get together and combine all their resources and just agree on a few animals to study. Now, in the last couple of decades, we've seen revolutions, and one of this revolution has been in sequencing technology. So whereas in the past, it cost huge amounts of money and a lot of effort and time to sequence the genome or figure out what the sequence was of all the genes in an organism. Now it's relatively inexpensive and does not take all that long. And so a lot of new animals have become things that we can study because we know what all the genes are. And then there's another new technology. It's called CRISPR-Cas9. And that's a technology that allows us to modify the genomes of not only the model organisms that people have studied for a long time, but these other animals that are much more difficult to modify. This is an example from my own lab where we've introduced a red color gene just to prove that we can into some of our epithelial cells. And so this technology is going to let us ask questions about what the genes are doing. We can tag proteins and watch them. We can mutate genes and see what happens if we get rid of a particular gene. So these kind of experiments are new and exciting and allow things like jellyfish, which are relatively understudied at the molecular and cell biology level, to all of a sudden be model systems for us.
Is the blue the nucleus of each cell?
Yeah, and these are these hexagonal epithelial cells like we saw before, and they're healing a wound. The green cells actually are expressing the natural glowing of these jellyfish. The red is a color that we introduced, and the blue is the nucleus in each one of those cells.
Neat. So what about in the ocean, Larry? How do you study jellyfish? How hard or easy is it to do?
Well, the problem is, of course, that they're fragile. Also, the problem is that sometimes they're kind of scattered. They're not very abundant in any one place. But to really find out what they're doing when they're alive, we figured out that we had to go to where they were. So we had to use open ocean scuba diving techniques that actually were pioneered back in almost more than 50 years ago by my professor at the time, William Hamner, where a bunch of divers go out in the middle of the ocean like this and are looking around for jellyfish. And when they find them, they've got a real high-tech system here of putting them inside a mayonnaise jar.
And that's you, isn't it?
Bringing them back to the lab. Yeah. And this actually was proved to be very useful because we could get live specimens. We could study them in the lab on the ship for a short while, learn more about their behavior, what they ate, what ate them, and so forth. So this really opened up gave us a vision into what the lives of these animals is like in their natural environment, undisturbed.
And Larry, I know that you're, along with being a biologist, you're also a terrific photographer, and you've taken lots and lots of photos of jellyfish. How easy is that to do in the water? Do you need special lighting, or how does that work? I mean, they're translucent, so. Yeah,
being transparent or translucent, they disappear into the distance. But if you have lighting that illuminates them from a little bit behind or from the side, you can get them to illuminate quite nicely. And they're really beautiful animals, as you can see. Looking at this, happens to be a pelagic snail, also a gelatinous form, and they really lend themselves to being photographed in situ. This is a sea cucumber, an animal that normally lives on the bottom, but there are pelagic ones that float up into the water, swim up into the water, and they are also very gelatinous. I mean, if you didn't know any better, you might think this was another jellyfish. These are salps, and some of the salps form long chains connected together, and some of them connect together little wheels like this one, where they're almost like segments of an orange, the way that they're fastened together, enable them to swim together. This one is called a doliolid, and it looks very much like a siphonophore in the way that it is structured, with many, many small units all connected together in a chain and pulled by a big, what you might think of as the locomotive at the front end, that swims along and pulls all these others behind it. And each of them is a filter feeder taking in small particles to eat. These can be up to 5 or 10 feet long. There's a tremendous variety of these gelatinous animals that have converged on this soft body, relatively efficient propulsion, and the ability to be either a predator or a filter feeder. This is an example of another siphonophore, which uses stinging cells like a jellyfish on its tentacles. It has a little red float in the center that keeps it floating upright. And siphonophores are made up of component pieces, much analogous to the doliolid that I mentioned, but the doliolid is much more closely related to human beings than it is to a jellyfish. It's a completely different organism.
Amazing.
Despite the convergence of form.
Yeah, very similar in form. Annette, I know you use some different techniques in your work to study jellyfish, including some genetic techniques, but different from the ones that JOCELYN was talking about.
Right, right. So we use a method called DNA barcoding, where we use a stretch of DNA sequence that can tell us what species that the animal belongs to. So we can take the DNA from the animal and sequence that barcode and identify it in that way. And we do this. This is part of our Ocean Twilight Zone project at WHOI, where one of the ways which we look at jellyfish in this region. The twilight zone refers to what we call the mesopelagic. It's that area between 200 and 1,000 meters water depth, and the sunlight at these levels is very, very dim. But it's a very important region because this is very critical to that carbon pump and Earth's climate that we talked about a little bit earlier. And so it's we want to know who these animals are that live there. But again, as Larry said, jellyfish can be very difficult to study. So we find that using genetic approach can be very helpful. And in particular, we're using something called environmental DNA, or eDNA for short. And this is a forensic type approach where we don't sample the animals themselves, we sample the water and we filter that water, right? And then we can basically collect the genetic traces that these animals are leaving behind. Think of it like their genetic fingerprints. And then we can get the DNA sequences from that, right? And then use that to identify the animals that live there. So it's really like thinking of it like a forensics type approach, where we don't even need to collect the animals for this approach. We just collect the water and look for those little traces that they leave behind.
Okay, so you're collecting the water, you're filtering it, you're getting the genetic code, you're reading that genetic code, and then you're comparing it to what? To a library of known animals? Is that,
That's right.
Or genes? Yeah.
Yeah. So there's a database that has reference sequences, right? And so that did stem from specimens that were identified, right? So ultimately, that is an important part of the process is having the specimens. So we compare what we get in our eDNA to that library or that database, and we look for matches for identification.
And when we're talking about the twilight zone, as you said, that's pretty deep in the water. So you're not diving down there to collect your water
Right.
samples. How are you getting those water samples?
Well, so you can get water samples through collection bottles, or we're using a new approach in combination with the eDNA. We're using a robot that called Mesobot that was we designed specifically just to study this midwater area and this midwater twilight zone area, and to study the life, ocean life in this region. And this is Mesobot, and it can track animals, it can take video images, and we can also, there's some video from Mesobot of a jellyfish, and we can also put samplers on Mesobot and to take the water samples. You can see those cylinders on the bottom part of Mesobot under the main body, and those contain filters that are collecting, filtering the water for collecting the eDNA.
I want to give a little shout out to Dana Yerger, who helped to was one of the leading engineers on Mesobot. I'm glad you mentioned Mesobot, because we actually have a wonderful children's book that just came out yesterday. It's called Where the Weird Things Are. There it is. And the main character in that children's book is based on WHOI's Mesobot. It's named Meso, appropriately, or Meso, I'm not sure how that's pronounced. And the story follows Meso as it dives down into the ocean twilight zone and meets all kinds of amazing creatures, gelatinous and otherwise, along the way. I think the illustrations in this book, I don't know if we have any examples, there we go. They're just beautiful. It's really well done. The story is really fun. And the other nice thing about this book is that the theme of the importance of diversity runs through the storyline, which is really great, both the importance of diversity in the ocean and ocean ecosystems, but by extension, the importance of diversity in human society as well. I encourage everybody to check out the book. It's available wherever books are sold, including our own local independent bookstore here in Falmouth called 8 Cousins. And we've got that link that you're seeing on your screen. And I'm not sure if we can put it in the chat as well, but the link on the screen will take you to the 8 Cousins website, and they can ship to anywhere. So please give them a look. I think we've got time to take some more audience questions. Ken, anything you want to share?
Yeah, sorry, you caught me when I was typing.
Sorry.
So, JOCELYN, we have a lot of people who are asking about the immortal jellyfish. Could you tell us a little bit about that?
Yeah. This the so-called immortal jellyfish is a jellyfish called Turritopsis, and let's see. And there it is. And I can just mention again about the life cycle of jellyfish, which Annette already explained. So they start out as a little polyp, and for certain kind of jellyfish, they spend most of their lives in the form of a polyp colony, and the polyps release the medusa, and the medusa come in male or female versions, and they can release either sperm or egg, and that sperm and egg fuse in the open water, make an embryo, and that embryo becomes another polyp. And what Turritopsis can do, it turns out, is if you poke it enough and damage it enough, the medusa kind of becomes a little blob, and then that blob turns back into a polyp. So that's been interpreted as the ability to actually go backwards in the life cycle. It led to people suggesting that it was using this term immortal to describe it.
Interesting. Anything else, Ken?
Oh, yeah. Let's see. For Annette, and I guess also Larry can chime in here, how many species are in your eDNA database, Annette? And Larry, how many species of jellyfish are there, and how do jellyfish relate sort of taxonomically to all the other sort of gelatinous animals that we're talking about?
Let's start with Annette.
Oh, sure. So I don't actually have the exact number. So I use a public database called GenBank, where people can deposit their DNA sequences. So when scientists write up their results and publish their papers, you make your data publicly available and you put your data on GenBank. And so we can we go to GenBank and we can get the sequences and use those to help us in our identifications. And one thing that we do find, especially, and I think it's actually true in coastal areas, but it's also true in the deep sea, it's that we need to keep building up our databases because they're not yet complete. They don't contain yet every single species that's out there, right? So we do need to, in parallel to doing the eDNA work, we need to continue getting the specimens and making sure our identifications are accurate and being able to identify them accurately, and then getting reference sequences. So we need to be continuing that work in parallel to doing the eDNA work. It's very important.
A little bit analogous to a global public library of DNA sequences.
Yes, exactly.
Okay, and then for Larry, tell us a little bit about how many animals we're talking about when we talk about jellyfish, and then when we talk about just the bigger category of gelatinous animals.
Sure. Yeah. So if we're talking about jellyfish, there are two major categories of jellyfish that are called hydromedusae and scyphomedusae. There's a few other smaller ones as well, but the total number of species is about 3,500 described species so far. So that this is the most numerous in terms of species of all these jelly animals we're talking about. The siphonophores that we showed a few of, there's about 175 species. The ctenophores, maybe about 200. So these are relatively small numbers. And of salps, only about 50 species. So some of these animals have what we call radiated into many, many more numbers of species than others. And the other thing that's interesting, of course, about them is that the jellyfish are a very, very old, evolutionarily old group of animals. There are jellyfish-like organisms in fossils in the Precambrian more than 600 million years ago. They've been part of our ocean as long as any other animal probably has been. Other ones are probably more recent origin, and even the salps, which, as I mentioned before, are more related to humans than they are to jellyfish because they are chordate animals, probably go back a few 100 million years. So most of these are fairly ancient groups of animals that have made a living in the ocean for most of its history.
That was actually a question I had for you, Larry. I was wondering if there have always been gelatinous animals in the ocean and how they originated.
Yeah, I think it may not look exactly like today's versions, but there probably have been representatives almost all the way back.
And JOCELYN, what about the use of jellyfish to understand human health or basic biology? Is that a new thing? Or you talked about some of the new technologies that have really advanced the science, but how long have people been doing that?
People have been studying jellyfish to understand jellyfish, of course, for a long time, but they've only really come into the lab as a model system, relatively recently. As a result, there's only a very small number of labs who use jellyfish as a model system. The jellyfish that I work on, Clitia hemispherica, I think there's now maybe 5 labs who study it. So quite recently, this is quite new.
We've got time for some more questions, Ken, if you've got some.
Oh yeah. For JOCELYN, is there any use, medical use for jellyfish venom?
I don't know a lot about jellyfish venom, but what I do know is that it's surprisingly complex, and I believe that the exact components of the venom are still not completely understood and are different in different jellyfish. Actually, I should probably refer this question to Annette. Probably knows more than I do.
Well, I think it's different for different jellyfish, but I can't answer the medical application one. Outside my area of expertise.
Oh,
But all jellyfish aren't venomous, or are they all venomous to some extent, but maybe not to us, for example?
So jellyfish are cnidarians, and cnidarians are defined as animals that have cnidocytes, and the cnidocyte is the stinging cells. So by definition, they all do have a sting. Go ahead, Annette. Sorry.
I was going to say, so the animals that we saw, like salps, right, and comb jellies, those are a different type of jellyfish. They're not cnidarians, so they don't sting, right?
Right.
So we want to distinguish between the cnidarian stinging types and then the non-cnidarians like the salps and comb jellies.
But
Even
with
among the cnidarians, the jellyfish which do sting, some of them do not have stings that affect us. So even though they're stinging, some of them are relatively harmless to us, where some of them, as you know, are quite harmful.
but fatal to their prey, presumably.
They are.
Ken, anything else?
Yeah, there are people also watching on YouTube. And Diving T-Dog asks, can you put a tracker on a jelly?
That's an interesting question. Larry.
Yeah, and actually, people have done that on a large jellyfish with a fairly firm bell, which is what we call that round part. People have put like a sonar type tracker on it, and the idea would be to see whether it's undergoing a migration, a vertical migration, day and night, or just to follow it around. So. It can be done. It hasn't been done very often, but people have been trying to do that with some success.
That's interesting. I know they do it for things like squid, but I didn't know they did it for jellyfish too. Ken, let's go with one more.
Okay, one more. Well, Amanda asks, and she says it's a silly question, which is always I love that preface. She asked, what is each scientist's favorite jelly?
I like it. Let's start with JOCELYN.
Well, let's see. I'm loyal to Clitia hemispherica, the first one I ever looked at, but I'm also very fond of moon jellies because I have fond memories of swimming among them in Woods Hole.
Annette?
This is a tough question too. So I actually used to study Clitia when I was a grad student and that family of jellyfish, but I also have become partial to the clinging jellies. Despite their toxicity, I find them really beautiful animals, and
I do too.
They're just fascinating to watch. Very mesmerizing, actually, to watch when they are pulsing. So.
And Larry.
Well, I'm going to have to go with the salps because I think they were the first thing that got me interested in this whole field of science many years ago. I always thought that they were fascinating creatures. The way they work, they're far more complex than the jellyfish. They have a brain, they have a heart, they have a more complex structure and behavior. And so, even though they sometimes come up in buckets full, like you see here.
Well, a bucket full of salps. It's amazing.
That's a bucket full of salps. They play an important role in the ocean economy, and they were neglected for many years. And I'm happy to have a chance to work on them for much of my career.
All right, I've saved a final. Just before we go on, I just think that image is so amazing. So the red thing is actually their stomach. You told me
that's their stomach. Yeah, and there's a little white thing that you see at the back of them. That's the place where their babies are attached. They produce little babies attached by a placenta, and that's the place where they grow from.
Okay. So I'm going to take the last question here, and I'd like each of you to tell us your number one reason why you feel that jellyfish and other gelatinous animals are important. Basically, this is a why should we care about jellyfish question, but from your own personal perspective. Let's go in the same order. JOCELYN?
Well, so I'm a basic scientist. I study very basic processes of how cells do things and what are the genes that control the things that cells can do and why we study them in jellyfish. I guess there's three reasons. One is to understand more about jellyfish, which I think, this audience likely will agree are fascinating animals just in their own right. Another reason to study them is to just get a better picture of the tremendous diversity there is in the way living things have solved common problems, like how to find food, how to perceive sensation, how to heal injuries. And the third reason is that as all basic scientists know, that you never really can predict where the next great discovery is going to come from that actually has profound impacts on human health.
All right, Annette.
So I think a primary reason to study them is to understand their functional significance in the ecosystem, their roles in the carbon pump, their roles in the food web, right? My personal motivation, I think, when I first got started with jellies was what still is, like, at the personal level, is the variety, the diversity of forms that they can take, and especially their life cycles, because they can have such complex life cycles, and there's so much variety in the life cycle. So was just really fascinating to me to think about how those evolved. And so that was that's what got me interested in them to begin with.
And Larry, you get the last word here.
Well, I think what I like about them is that they're extraordinarily diverse. They're extraordinarily strange and beautiful, and they're unlike anything that lives on the dry land. And if you think about how big the ocean is, these are animals that live in the biggest living ecosystem anywhere in the solar system. And they've been there for the entire history of the Earth, and they are kind of masters of their universe.
All right, that's a great closing line. So we'll leave it there. I want to say a big thank you to Annette Govindarajan, Larry MADIN, and JOCELYN MALAMY for joining us tonight to talk about jellyfish and other gelatinous animals and their connections to the ocean and to human health. Thank you also to my WHOI colleagues who work very hard behind the scenes to make this event possible, and to everyone who joined us on Zoom, Facebook, and YouTube. Thank you. Tonight's event was the second in our 5th season of WHOI's Ocean Encounters events. Please mark your calendars and join us again on April 12th for our next Ocean Encounters. We'll be talking about plastic in the ocean, where it is, how harmful it is, and how we know. Please watch our website, whoi.edu, for more information about that. In the meantime, you might also want to take a look at the very cool merchandise at WHOI's online store. That's shop.whoi.edu. You'll get a 15% event discount for joining us tonight. Just type in the discount code JELLIES. If you enjoyed tonight's Ocean Encounters program, you can be part of WHOI's solution-based approach to ocean science. If you're already a WHOI member, I want to thank you very much. If not, please consider joining. You'll be providing crucial support for our outreach and our science, and you'll get some great benefits in return. Memberships start at just $40 a year. The average gift is $120, but any amount helps. Your support will make a difference. If you can, please stay with us. We've got a great 4-minute film to share with you. It features Larry and Annette, actually, and you'll learn more about jellyfish, the robot Mesobot that we talked about earlier, and even get a bit of a look ahead to our next event about ocean plastic. In the meantime, on behalf of Woods Hole Oceanographic Institution, thank you and good night.
Jellyfish, or things very similar to jellyfish, have been here in our oceans for more than 600 million years. They're part of the history of evolution of life on Earth. A lot of them live in this twilight zone area that's down to about 3,000 feet deep. And we don't know very much about anything that lives there, let alone the jellyfish. Sometimes we call that inner space. It's like things that would live in outer space because it's so big, except it's on our planet, it's all water, and it's just totally different from what we're used to on land. You know, jellyfish had been studied for 100 years, but it was just jellyfish that came close to shore and they were easy to catch and that people knew about. And now that we know that so many of these jellyfish are out in the open ocean and the deep ocean, we need ways to get there.
Well, the Mesobot is a new kind of robot specifically for exploring the twilight zone. Its job is to emulate many of the qualities of a skilled scientific diver. Being able to hang almost motionless in midwater and observe the animals, to take samples, to take measurements, and to do that in a very unobtrusive way.
We've been working on an environmental DNA sampler that goes onto Mesobot. eDNA refers to pieces of DNA that's in the environment that were shed by the different animals that live in the environment. So it's pretty exciting in that we're actually, with this eDNA approach, we're not even sequencing the animals, we're sequencing their forensic fingerprints or their traces of DNA that have been left behind.
When we say we're worried about the future of the ocean, it's not that the ocean is going to go away. It's that we're worried that the ocean, which has been a beneficial part of human life and human evolution, is going to change in ways that won't be so beneficial anymore. People have found plastics almost everywhere in the ocean, including the deepest parts of the ocean, like in the trenches. So we know that the plastic comes in at the surface or at the edges of the ocean, and we know that it's showing up at the bottom. So it has to be in the middle in some parts of the twilight zone and in the deeper part of the ocean. A lot of different kinds of animals we're seeing plastic in their stomachs now, and fish and marine mammals and turtles and so forth. And so a lot of these are things that feed on jellyfish also, and so easy to mistake the jellyfish and the plastic. So I think that that's the real concern right now, is the larger things which just look to a predator like something that they're used to eating, but they're not.
Understanding the twilight zone will be a key part of managing the oceans in the future. We believe most of the fish in the ocean live in that region. If we're going to protect it, we first have to understand it.
Even for our coastal jellyfish species, we don't have a good handle on who they are. And so that makes me think, if we don't have this knowledge in these, quote, easy places, what does that say about somewhere like the mesopelagic zone, where we've explored such a tiny fraction of it? What's waiting there for us to discover?
I always like to find new things, new species, and the twilight zone is a great place for that. That's one of the coolest things, is you go there and you find something that's probably been living there for millions of years, and you're the first person to see it. Doesn't get much better.